that's present within the small intestine produces a hormone called cholecystokinin. It's oftentimes abbreviated CCK. And I'm going to spell it. CHOLECYSTOKININ. Cholecystokinin. It's very analogous, this situation is, to the enteroendocrine cells that sit within the stomach. When either protein or fat enters into the small intestine, these enteroendocrine cells release cholecystokinin. And what cholecystokinin is going to do is three things. Okay? It's going to stimulate the gallbladder to contract. Okay? So that bile is excreted through the cystic duct into the common bile duct. So number one, it stimulates contraction of the gallbladder. Number two, it stimulates pancreatic secretions. So at the same time we're mobilizing bile, we're also producing pancreatic secretions containing enzymes. Okay? And these are going to travel down that major pancreatic duct. And then the third thing that cholecystokinin does is it stimulates relaxation of the sphincter of Oddi. So that these secretions, the bile and the pancreatic enzymes, as they're passing into the ampulla vata, then they're going to pass through into the duodenum, where they can interact with the small intestinal contents. Okay. And what we'll see later is that the bile and the pancreatic enzymes are going to be necessary for the breakdown and absorption of fat. The pancreatic enzymes are also going to be necessary for the breakdown of proteins. So it's important that when these nutrients reach the small intestine, that these substances are released into the lumen.
Are all enteroendocrine cells paracrine?
No, that's an example of a hormone. That's
Right.
not a paracrine hormone, that's a traditional hormone. It's released into the bloodstream.
Okay.
It doesn't act locally, it acts at a distance. Okay. I'm going to mention something else, is that when you look at the epithelium, okay, you may see occasionally lymphocytes superimposed. Those lymphocytes are coming from the lamina propria. The lamina propria in the GI tract has a lot of lymphocytes. Sometimes they're organized into bulk, lymph follicles, but oftentimes they're simply scattered lymphocytes within the connective tissue. If these lymphocytes then move from the lamina propria into the epithelium, it may look like you have a stratified epithelium. But remember that these are not fixed cells. They're not tethered to a basement membrane. They just happen to be superimposed upon the epithelium when you're looking at that tissue. All right, so what we see here are villi. This is the lamina propria. It's got lymphocytes. That's what those purple dots are. Now, as we come down into lamina propria, these are crypts of Lieberkühn, or intestinal glands. Okay? The reason you're seeing them in cross-section here is that as these tubular glands move distally down into the lamina propria, they coil. And so when you cut the tissue, you may cut part of those glands in cross-sections. This light staining region right here is your muscularis mucosa. So anything below this, in this area here, that's your submucosa. Okay? Now, above the level of the submucosa, if you look at this tissue from here on up, the only thing you can say is that that's small intestine. You use the submucosal contents to differentiate duodenum from jejunum from ileum. The duodenum is the only part of the small intestine that has these glands. These are light staining, mucus secreting glands within the submucosa of the duodenum. They're called Brunner's glands. So this is an identifying feature of the duodenum. So without the Brunner's glands, you can say, well, this is small intestine. If you see Brunner's glands, then you're more specific. You say this is duodenum. Now, deep to the submucosa, you have the traditional muscularis externa. Intercircular, outer longitudinal. Okay. And then finally, if we're talking about duodenum, you're more likely to see an adventitia. In the jejunum and ileum, because these are intraperitoneal organs, you see a serosa. Okay. I'm going to mention one other cell type that we see along the deepest part of the glands. Paneth cells, P-A-N-E-T-H, Paneth cells. These are cells that have a lot of secretory granules that contain lysozyme. Lysozyme, you may remember from microbiology, is an enzyme that breaks down the cell walls of bacteria. Okay, they break down peptidoglycan. And so this lysozyme helps to protect against bacteria that may be trying to invade. So it's part of our innate immunity. So along the very base of these glands, you can see the deepest part of the glands, you may see Paneth cells. We have some slides that are specially stained to identify those Paneth cells. All right, we said that to differentiate the duodenum from jejunum to ileum, you have to look at the submucosa. We said that what identifies the duodenum as duodenum is the presence of Brunner's glands. Jejunum is identified by the absence of Brunner's glands and the absence of what are called Peyer's patches. Peyer's patches is going to be a feature of the ileum. So P-E-Y-E-R, ''S, patches. Peyer's patches are simply collections of lymph follicles within the submucosa. Sometimes you'll see these lymphocytes extending upward into the lamina propria. But within the ileum, they're localized mostly within the submucosa. So if you see Peyer's patches within the submucosa, then you've further differentiated that small intestinal segment, and you say that is ileum. If you see neither Peyer's patches nor Brunner's glands, then that region of the small intestine is the jejunum. Now, you'll notice that the morphology, the shape of the villi is a little bit different as you go from duodenum to jejunum to ileum. Okay? In the duodenum, these villi are somewhat broad. They can be slightly irregularly shaped. But when you reach the jejunum, they become taller and thinner, a little bit more crowded, as we see here. You can see the intermittent goblet cells. Okay? So in the jejunum, the villi are taller. Again, they're a little bit more crowded. And by the way, this is jejunum. You can see here's the muscularis mucosa. Here's the submucosa. These are only blood vessels. These are veins in the submucosa. But you don't see any Brunner's glands. You won't see any Peyer's patches. If now we move into the ileum, okay, you see these collection of lymph follicles, which identify this region as ileum. In fact, you see germinal centers here, which tells you that these lymphocytes have been exposed to an antigen and are responding. These Peyer patches are also part of MALT, mucosa-associated lymphatic tissue. The villi within the ileum tend to be branched and tend to bend a little bit, or coil. For that reason, they're often broken up when that section is cut in a plane. Okay, so many times in the ileum, the villi aren't as nice, okay, as you would see perhaps in a duodenum jejunum. They kind of cut in oblique section or cross section, so they're in pieces. Not all the time, but many times that's the case because of the fact that they're branched and they tend to bend or coil a little bit. Okay, so this is the submucosa. This would be the muscularis externa, and out here you would have a serosa. Let's take a 5-minute break.
So it's not in the cavity as adventitia. The ileum is more anterior, so it has a serosa.
Yeah, the duodenum and the pancreas have adventitia. Also, is obviously like the esophagus because it's not in the stomach yet.
This was the site follicle, right?
Yeah, that's just the lymph follicle.
Where was this found?
That's in the esophagus. You see, you have isolated or scattered lymph follicles throughout the GI tract. In the ileum, they cluster.
Okay. So that's the only place where they're very close together.
Now, you will also find that in the appendix.
Oh.
Anytime you have an interface between the outside and the inside world, you see collections of follicles or lymphatic tissue. The tonsils form a ring around the pharynx. Why? We're protecting inside from the outside. So what these follicles are doing is protecting the small intestine from bacteria that may be coming in from the colon. Because the colon is chock full of bacteria.
Yeah.
And if those bad boys want to invade the wall, they're ready for them.
So you'll see the germinal center in any of those areas if it's been exposed to an antigen?
Yeah, the germinal center is just proliferating B cells that have been exposed to an antigen.
What did you say the lacteals?
The lacteals are for the absorption of fat.
It's cirrhosis.
I don't have it with me.
Okay.
I can tell you what you
Dr.
got.
Finner. It is. Why?
Dr. Finner.
What is it?
1 second. Okay.
I'll write it down.
So,
We're talking about the cirrhosis. But it was in the cirrhosis on the
yes. Who's that?
She wants to know if there's a muscularis serosae.
Is
Say it
it?
again?
Is it?
A
Because me
muscular
and Kat.
serosae.
There's no muscular.
There's no muscularis.
Then what is it?
You have a
Muscularis
muscularis
mucosae.
mucosa, which is part of the mucous membrane, and then you have a muscularis externa, which is just deep to the outer surface of the wall.
Oh, okay.
But serosa is on
Yeah, the throat is on the outer surface of it, it's the surface of the wall.
Okay.
I'm having the hardest time distinguishing the skeletal from the,
Smooth muscle.
yeah, in those sections, like I
It's
said.
very easy in cross section, like we get on the slide. In cross section, the skeletal muscle fibers have a lot larger diameter. Smooth muscle fibers are a lot smaller.
Okay.
So it's really easy.
Because I guess I'm trying to look at like the nuclei, if they're, in
Well, that's the other
line.
thing, is that in skeletal muscle, go look at those cross sections. The nuclei are peripheral. In smooth muscle, it's not only that the fibers are smaller, but the nuclei are centrally located.
Time change.
You haven't acclimated to it.
Dr. Karen, do you, schools open this Saturday?
I don't know.
Because I saw on the board downstairs by the cafeteria or whatever that, it said school closed and it's like arrow starting on Mondays, like, can you get something?
So then it would be open on
I'm
Saturday.
not sure, but I didn't know if you knew.
Yeah.
So that makes it so much easier. The first 11
We're gonna be off all next week.
When's our test?
Like,
The Wednesday after Saturday.
Is it
Okay, so
first?
it's Wednesday after we come back.
Yes.
Is it April first?
So are we so we're dissecting the dog on Friday?
Well, I'm trying to think. I think what we'll do,
Come on, it's
We haven't even gotten through the whole thing yet. Wait.
I think we'll push the test back to Friday.
Okay.
Thank you.
Isn't
Wait,
spring
what's the
break
date?
next week?
Friday
So
after
we have
spring break.
2 more days.
What's the date of the Friday?
Friday is April 1st.
Okay, I thought that's what it said on the schedule.
Why does my jacket
Well,
smell?
so-
I had it done.
Why did you just do that?
So we're
Cold,
moving it to
shivering.
Friday?
Yeah, because we do have to dissect the dog.
Yeah.
You know, we have to cover nutrition, we have to finish this with slides. There's a lot
Those are
of
awesome.
things.
They were better when they were
While we're
69
doing
cents
this,
and $1.29.
Oh, sure.
Does
Yeah.
that happen with the rat?
Stone ground.
A rat?
A rat, yeah. It did that to me.
A
Well, not to
rat?
me, but a
Wait.
friend.
What? So
You don't have to worry
We're
about
not
too much
redissecting
splash
the dog?
with the rat.
Yeah, it did it to,
one's a flip test.
You know, we had a student,
Oh,
you
God.
know Arden?
out there.
yeah.
Okay. She works at Turo. So she took histology, and we had a special project at the end of the semester. Her and her partner caught a mouse, preserved it in formaldehyde, brought it to class, dissected it, so took slices, biopsies from all the different organs, brought it to the Turo pathology lab, created paraffin blocks for all of the different sections, cut it up, created a slideshow where they prepared not only slides, but a PowerPoint demonstrating all the features of all the different tissues from that mouse that they caught.
That's super cool.
Whoa.
Yeah.
Who got time to do that? That's a lot of work.
But it was
Talk
a
shit about
learning
art.